we were still going through the introduction, which is fantastic. It means that we're actually continuing to interact. We're supposed to start now, right?
Yes, that's right.
Oh, just dawned on me. Am I starting too early? I don't do well on time. Another little story about me. When I first started my job, and I was trying to impress everybody with my teaching back in 1989, and it was going okay. But they sat in, the senior faculty sat, 3 of them would sit in many of my classes during my first few semesters of teaching and would evaluate me, which was good. They gave me feedback about how to do things. And I did the same for junior faculty and still do now. But I think it was my second semester, and I asked the students, I forget time a lot of times, like when things are supposed to end, because I get to talking about this. So I was in this huge lecture hall, 300 students or whatever teaching, and I asked them on like the second day, I looked at my watch and I said, what time are we supposed to end? And they told me, and I said, oh, okay, thank you. Anyway, and the senior faculty came in to evaluate me a couple of weeks later, and one of the senior faculty members came up later after the class and said, your lecturing is really going well. You're doing a great job of interacting with the students. We're really proud of that and really pleased with that. But why are you letting them out 15 minutes early every time? And I said, I am. And I went back in the next time and I said, I should just give every one of you a 0 today. They giggled. They knew that I wouldn't do it to them, but I was like, really? Seriously? Thanks, guys. Okay, good. That's all. All right, so I would like to ask, once again, if you have questions, we'll continue on with those. If you do, I don't have much more on this, but if you have questions for me, continuing on from where we were, because we had to break for coffee, tea, etc. Anything else? Yes?
Since you spoke about adaptive radiation and since you cited fishes from the African Great Lakes, I wanted to ask you, if you look at adaptive radiation across the globe and look at all the examples of adaptive radiation across different taxonomic groups,
Yeah.
do you always see that these have been a result of diversification as a result of genetic exchange as a, you know what I mean?
Yeah, oh yeah, I do. Yeah, no, so this is going to be another one of those times where we have to say, I'm not sure yet. What I can tell you is that Ola Seehausen in 2002 or whatever in a Trends in Ecology and Evolution paper, it's cited in my book, Ola Seehausen, Seehausen, if you want to look it up. But he proposed the hybrid swarm hypothesis. And a hybrid swarm, if you don't know what that is, it's just like it sort of sounds. You get genetic exchange, and he was talking about eukaryotes. So in our plants, we have a red flower and a lavender flower. They cross in nature. They form an F1 that's sort of a wine color, red wine colored flower, really beautiful. And then if it crosses back to the parents or if it selfs, you get this enormous array of floral colors and different heights of flowers and leaf shape and all this sort of thing. That's a hybrid swarm, okay? So an early hybrid generation, really diverse phenotypically, broadly morphologically diverse, and also genomically. Okay, so he proposed this in this review paper and said this fits what is seen in Darwin's finches, the Rift Lake cichlids, silver sword plants in Hawaii, and he named a bunch of different kinds of groups where they knew that there was basal intergressive hybridization that had gone on, or hybrid speciation. In the case of the silver swords, they're diploid on the mainland of North America, and they're all allopolyploids on the Hawaiian Islands. So he proposed this model and said, now here's how we have to test it. And so what they've done, he and a number of other people have been testing this model in various groups. And so we now have a few examples, a handful of examples where we can definitely say that at the base there was intergressive hybridization that was introduced into an environment, or that there was repeated intergressive hybridization like in the Darwin's finches, or there was repeated intergressive hybridization, adaptive integration by the looks of it, in canids, dog-like things. So you notice that most of the examples I just mentioned are animals. And so the evidence is building that it may very well indeed take that many kinds of mutations together to give you the kind of explosive radiations we call adaptive radiations. But I cannot say that every adaptive radiation we see out there is due to genetic admixture. If you wanted to ask my gut feeling, I think you have to have that kind of phenotypic and genotypic space to begin with to get that many new lineages over that short of a period of time very often. And Heliconius, of course, in Central South America is another example. I mean, there are just many, many examples where people know, yeah, it's underlain by shuffling genes around. But I don't have an answer of what about rhododendrons in China. They were asking me about that. You know, what do you think? They asked me specifically I was just at Guangzhou at the South China Botanical Garden, and we were talking about this, and I was meeting with some of the scientists. They said, we have this explosive radiation. There isn't a lot of intergressive hybridization in rhododendrons. We know that. So do you think our explosive radiation was due to natural hybridization? And I said, I hope so, for my own interest. But I said, I don't know. You have to test it. You have to go and do the kind of timeline and ask where you had those and what kind of changes biogeographically we think might have been there. It's easier with the Rift Lake cichlids, right? Because you have a contained unit in a lake that you know wasn't there 15,000 years ago. And now you have 600. So there was good, so anyway.
So every instance where they have tested it, they have found evidence in favor of
Yeah. Yeah, well, yes. They have, see, but here's the deal. We're talking about two different things, right? Is there good evidence for genetic admixture, not even in adaptive radiations, but let's keep it there. Is there good evidence for genetic admixture during the evolution, say, at the base of an adaptive radiation where now you have 600 things? The answer is yes. They have good evidence of that, but it doesn't mean it's causal. Right? It doesn't mean that it's the reason you have adaptive radiation. Yeah, you had a lot of phenotypes and genotypes. It makes sense. It's intuitively, speculatively interesting and correlatively very interesting and fascinating. But what you actually have to do is ask what genomic and phenotypic variability was circulating in that group of individuals, and how might that have led to what you see now as those 600 different phenotypes and genotypes. And what they've done in Lake Malawi, for example, I just reviewed another manuscript for somebody who has to remain nameless right now for science. I think it's going to get in there. And they have done such a really nice job of tying what the allelic variation was in the female's opsin genes, female fish's opsin genes, because they've inferred sexual selection as a big driver of that adaptive radiation, how females perceive males. And so her opsin gene alleles, depending on which lineage she's in, it shows an enormous amount of allelic variation that was brought in due to intergressive hybridization, as was male coloration. So they have they've done the experiments and tanks and all this sort of thing, so they have much better information of what could have caused it. Why intergressive hybridization and that admixture might have been what seeded that lake to split them out into 600 different lineages eventually. But so having the hybridization down there, quote unquote, at the base of a adaptive radiation is only one small piece of it's an important one, but it's only one piece of the puzzle. Because you really have to get to causality. And all of you know, to get to causality in ecology or evolution, because it's always retrospective. Well, ecology, we do a lot of experiments, but still, in terms of evolution, it's always retrospective. You're always looking backwards and then trying to get the strongest inference. So, yeah.
Are you saying that hybridization itself might not be the causality, because that itself can give you adaptability to new ecological habitats and
Right. Yes. It could. Look, you're talking to somebody who really actually infers that that's what's underlying most of these adaptive radiations, if not all of them. Okay, that's my assumption or inference. But what I'm saying is to actually, for someone who's not, and they say, well, yeah, you had hybridization down there, and yeah, you had hybrids that got into the lake, but so what? It's neutral variation, probably. It's going to sort out in the environment. It's going to be lost. It's not who knows if it's adaptive? So you have to actually test why the adaptive radiation might have resulted from the genomic variation and phenotypic variation that, yeah, was there from hybridization. But what if it's all neutral? You know, what if what if the male coloration doesn't make any difference? What if the opsin genes to those fish now? So that's why I'm saying it takes explicit and very time-consuming and really expensive in a lot of ways experiments, but you have to do those to actually then say, yeah, this is why we think it caused it. Yes, it was there, but this is why we think it actually caused it, was adaptive. Like I say, I'm having a hard time not just saying, of course it caused it. I mean, how else would you get that kind of genotypic and phenotypic variation that you needed? We couldn't get that many point mutations that quick, but that's not science. That's just my opinion. For me to say that. Okay, what else? We'll be talking a lot more about, obviously, these topics you'll get really tired of this probably. But the nice thing about it is you have other people talking about really fascinating things in between when I'm speaking about things, so you'll get a break from me. No, it's true. It's a good thing. Once again, ask my children or my wife. My wife, who just texted me and said that she just had a foot massage, which she thought was a foot massage until she found out that they walk all over you. She loved it, by the way. Hey, she's going to try to talk me into one. I absolutely know it, and I'm going to go, I don't think so. Students will probably want to do that by the end of the course, but I'm not going to do that. All right. So I mentioned that, and if, once again, questions, just interrupt. So we're going to talk a lot about us and our associated organisms to use to illustrate evolutionary diversification accompanied by genetic exchange. And for example, I'll give you this example again. I'm not going to show you all the data, but Anopheles mosquitoes right now, because of various places we're traveling, we're on anti-malarial drugs, which we take occasionally. And so, we worry, this is a part of our ecology, right? The disease vectors, the Zika virus in South America that people are worried about from Africa and that sort of thing. That's a part of our ecological setting. And I'm really going to emphasize this a lot with you, is that humans have an ecological setting. I know we're sitting in a room that's cooled right now. So we modify our environment greatly. We purify water. We have drugs to take for diseases and things. But we still have an ecological setting, right? We wear clothes, we eat food, blah, 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 and we battle things like this. And so, this is a panel I love showing still. Our drugs. And Frances, my wife, always points out that this is her drug of choice, chocolate. And these are my two, espresso and red wine. But every one of these, and neither one of us has actually ever tried this, so I don't need any hands up about that one. But every one of these products, and in fact any drug you can think of, let me put it this way, I have never found an organism that we are associated with that's a part of our ecology, whether it was food, whether it was clothing, whether it was building materials, whether it was drugs, that is not a part of the web of life. I have looked for them because I want exceptions, but I have not found a single one. It's hard to do. It really is actually hard to do, and that's because we're finding out of how organisms diverge, but still. But, you know, for example, the red wine that I like a lot, evolutionary advantages conferred by an eukaryote gene transfer event in wine yeast. In fact, all yeasts that we use for fermenting things, whether it's alcohol or not, all of them are able to do that because of transfers that allow them to live in really hostile environments. Elevated alcohol, whether it's fermenting bread or whatever, elevated alcohol, sulfides, all sorts of things that should kill them. They've taken in genes, we didn't give them the genes, they've taken in genes so they could actually survive. So it's that sort of, you know, web as well. And of course our own species demonstrates these kinds of complexities. And we'll talk a lot about this. The old, and I emphasize that because it's ironic, 2014, I'll show you another one that's 2016. I don't know if it's now though. So the old one, which is much less complex than what we know it already now is, you know, that we were hybridizing, our species was hybridizing with other species within Africa before we ever left, moved out. Then we had admixture integration with Neanderthalensis, something called Denisovans, also with other kinds of extinct forms of our genus, and extinct forms within our genus were also hybridizing with one another. We've detected that now. And so those are the kinds of topics, I mean, we're already discussing these. Those are the kinds of topics that we're going to continue on with, okay, as we talk today. Today we're going to focus actually now, the readings, as you will have noticed, are actually on classical, as we went through earlier, they're classical readings on what we call speciation. But they have incredible implications and incredible things to say to us about what we've discovered since all of those papers came out, all the books, Darwin's, etc. We now know once again that organisms don't diverge the way we thought they did, you know, in geographic isolation and that sort of thing. But that doesn't make the papers we're going to talk about now that I'm going to open up the floor to any less important. Okay? All right, questions or comments? Any more questions before we start talking about the readings? Yes?
Since we are talking so much about genomics, I would like to ask you what is the role of environmental factors affecting speciation?
Yeah.
So we have a phenomena or a concept called phenotypic plasticity, where if you have variation, you have standing genetic variation in the organisms, and because they face certain climatic or ecological variables, they have different mobs. So these mobs, if they are adaptive or if they are selected for, are they capable of giving rise to a new species? I mean, does that also play a vital role in speciation?
Yeah. So the question is, if you didn't hear it, is what about the role of basically G by E, but environmental selection, ecological selection? Because I'm a pan-selectionist, pan-adaptationist, it's sort of central for me. So when I think about speciation, biodiversification, adaptive evolution, I'm automatically thinking about environmental selection. And so I have to be careful because you know, sexual selection needs to be taken into consideration, and I then think, well, that's partly environmental depending. But so In terms of plasticity and in terms of environmental selection just in general, I think it's central. I think Darwin was right. I think natural selection really is the driver of evolutionary diversification. We will talk about how that can go hand in hand with something like genetic drift, but I still maintain that all the evidence that we have when we do experiments in the field, I don't mean just us, but people across evolutionary biology, evolutionary ecology, tells us that environmental selection is central. Can phenotypic plasticity, those that because of environmental shifts, can phenotypic plasticity lead to new lineages? The answer is it depends on the genomic underpinnings and the phenotypic underpinnings. The theory would tell us, but I would argue, yeah, absolutely. And we're going to talk, I'm actually going to give you a talk that I was thinking would be a public lecture here on rapid evolution due to genetic exchange kinds of events. And it's that, the question is, in our climate change environment right now, climatological rapid evolution that we have going on right now, can organisms keep up?
So, is it possible under common garden experiments to demonstrate this? I mean, if we just take two species and treat them in separate conditions for generations, in that case, we are actually separating the species from its actual wild type conditions.
Mm-hmm.
And so there are no interactions whatsoever with the nature. And
Yeah.
then if we demonstrate something like that, is it possible that we could actually conclude these things? I mean, it's just a doubt whether common garden experiments can be used to demonstrate the separation of these species.
Yeah, so it's a good question. What is the utility of common garden experiments or cage experiments or greenhouse experiments or whatever? And what I would suggest is that if we're only using, and we've done this, if we use greenhouse experiments, for example, and put them into our plants into shaded to see whether or not one of our species is shade tolerant versus the others or whatever it happens to be, I think we can get a hint of what may be going on out in nature. It can give us a way then to take, and I'll show these data when I'm talking about our plants and ecological adaptations. It can give us direction in what we might want to look for in nature. And when I say common garden, what I actually am talking about is actually taking genotypes and placing them into different environments in nature rather than like an experimental garden per se. So when we do, that's the way we do them anyway. And there are good reasons for not doing them that way. I mean, there are other reasons for doing them differently. But we actually take our genotypes, and we take exactly the same genotypes because our plants are clonal. And so we create genotypic masses, and we break pieces off, and we put them in exactly replicate them in different environments in nature. So I'm thinking of them in that way to see who's more fit in what environment and to try to get an idea of why. But I think in that way you can do it. I think you can tease things apart. However, I don't think we can. I know we can't do that in the greenhouse. It's absolutely impossible. But the greenhouse experiments can be incredibly important and powerful to tell us what to look for in nature. If we hadn't known that one of our species really couldn't exist in water, then some of our natural experiments wouldn't have made any sense, for example. But it's a good question. How do we do this stuff? How should we test our hypotheses? Sometimes it's just we can do it this way and we can't do it that way. I mean, if you're working on animals, it's real hard to do a natural field reintroduction common garden. They tend to leave when you put them on the ground, No, I mean, there are ways. I know I'm being a little sarcastic, but I mean, I had a colleague who actually set cages out in natural settings, and that for insects, by the way. It wasn't like tigers. But anyway, so they, put them out there and was able to study them. I wouldn't have wanted to have done it. Great. Other questions? I'm not ignoring you. I'm just going to put this down and pop something else up. Anything else? Till we... Okay. All right. I've given you the introduction to the course. You guys are doing fantastic. I really, really have good interactions in China. I have before I say what I'm about to say, so many good colleagues there, so many good interactions. I actually have a partial appointment there called a 1,000 Talents Grant appointment. So and my daughter-in-law is Chinese, so I have nothing against the Chinese. But boy, was it was so hard to get them to talk and ask questions. It's a language problem because I don't speak Mandarin or Cantonese. So it's my fault, but still, you guys are doing great. This makes it so much easier. Thank you, is what I'm trying to say. All right. I'm going to ask you a question first, though. So here are how do we define species, right? That's what we're doing here. It's called species concepts, but how do we define species? And let me ask you a question. I've done this, finally. Chapter 14 is from Origin of Species. Why did Darwin write Origin of Species? Let me ask you another question first. Do not be embarrassed. I did not read Origin of Species until I was a junior faculty member, okay, literally. That class I told you about, I was reading Origin of Species. That was the first time I'd ever read I'd read little bits of it. You know what I'm about to ask. So from beginning to end, who has read the entire Origin of Species here? Anybody? Shame on you. What awful people. No. I would encourage you. Look, I know, who am I to say, hey, add several hundred pages of more reading onto yourself. But it is the foundational volume about evolutionary biology. And it's really well written. There is a reason it is a classic book, and a bestseller. It's not just the ideas. He actually wrote real well. It's a little flowery in places, but it's actually an excellent book. Why did he write it? Do we know that? What were his reasons for writing Origin of Species? This will have to do with it. We'll get to what he said about species concepts. Why did he write Origin of Species? Anybody? Yes.
Because his mayor wrote a letter to him about that the population, something like that, population
Okay.
statistics. So this made him inspired from that, gives knowledge to the world.
Okay. So he was prodded, right? Charles Darwin was prodded by various people. What were his goals for writing it, do you think? You touched on it, yes.
I guess he wanted to speak the truth about what he had found out, that species were not made as they were, but rather are there now as we see them because of variation.
Mm-hmm.
This continuous variation, I guess, he found out that it led to species, and species are not formed as they are.
Okay. So there were two models. There was a main model held by scientists, not just non-scientists. There was a model out there about how species came to be that he wrote in opposition to. What was that model? Creationism, right? And I emphasize that scientists, and I'm talking about biologists here, biologists, geologists, etc., held to a creationist model. And so he wanted to demonstrate first, one of his main goals was to amass an enormous amount of information, of data from different kinds of studies to indicate that instead of special creation, every species being created independently from every other one, that actually what had happened was that you had what he called descent with modification, right? Evolution, what we call evolution. Okay, so that was his first goal. What was his second goal? Major goal.
He was trying to explain the diversity how all this so many.
Okay.
And he came up with the explanation for the mechanism of evolution.
Which was,
Natural selection.
okay. And so his two main goals then were to counter creationism. Give me a second and I'll be right back with you. Okay. Stay. So, he wanted to argue or counter creationism, or what we call today special creation. By the way, 50% of the, 30 to 40% of our National Academy members in the U.S. are creationists. 50%, at least 50%, don't laugh at us, at least 50% of the students that I teach in my undergraduate evolutionary biology class will be creationists. And so, see, I'm Christian too. It's an interesting mix that I'm an evolutionary biologist and that as well. And so, this is a huge topic, huge debate for us. You would think that it would have been over in 1859, but it's not. But this was one of his goals, and we'll talk about that in a moment. So he was supporting evolution, but he proposed the mechanism, as you said, he proposed the mechanism of evolution to be natural selection. This was not completely novel. You probably know this. I mean, even his own grandfather had drawn up evolutionary trees and had proposed evolutionary mechanisms. So Darwin was not, as sometimes you get this sort of cartoon or whatever they call them now, story that Darwin went out on the Beagle and he was completely naive to evolution. He was just complete creationist and never heard anything about evolution. That's never the case. It's always more complicated. Was he a creationist on the Beagle? Reading his Voyage of the Beagle that he published in 1846, I would say yeah, he was pretty much still a creationist, still held to that model. But when he came back, he had been definitely impacted by what he'd found. But this was not novel. What he did here was not novel. It had already been around, but it had never been done the way he did it, which was to amass so much information to point to the reality of descent with modification. No one had ever done that. This was novel. You know, where he got the idea from artificial selection, etc. Okay, so what was Darwin's species concept? I gave you the last chapter in Origin of Species. This is not going to be easy to have gotten from this chapter, but if you read the whole book, it wasn't easy to get it either. What do you think his species concept might be?
The idea was the continuum. He had an idea for species continuum.
Mm-hmm.
What he proposes, why we see the species that we see today is because the ones that do not exist anymore, they were not able to survive. They had a geofitness at some point of time and they dropped out. That's why we see distinct entities as species.
Mm-hmm. Okay. And so if I gave Darwin because someone did this one time. If I asked Darwin, how many species of Louisiana irises do I have? How would he have told me, do you think, to diagnose how many species I was looking at? Because that's really what we're saying, right? It's like, how are we going to define in this group of plants or your group of animals, how would Darwin have decided how many species you had? Yes?
I'm guessing based on morphological distinctness and reproductive isolation.
Okay. And would he have said, absolutely, he would have used those kinds of characters. If he had genomics, he probably would have used those. Given that special creationists in his time and before had been arguing that species were created with each individual species had special attributes, special creation. Each were inherently special. Is it any surprise to you if I tell you that Darwin didn't have a species concept? The author of Origin of Species suggested in no uncertain terms, he wrote, species are like genera. They're artificial constructs of humans. They're not real entities. They're not real biological entities. That doesn't mean that he didn't name species or decide how many species of barnacles that he had. He wrote a whole book on that, by the way. Okay? It's that he felt like, in fact, someone asked him one time, like I posed, how many species of Louisiana iris are there? And the group that they were being asked about, he said, I'm not an expert in there. Go ask an expert. They'll be able to tell you, but species are not real. But do you see why he would argue that philosophically? I mean, his book was called Origin of Species. Why would he have not really defined how to define species?
I think also he recognized
Whoops.
the fact that it was very subjective and depended on the taxonomic expert of that particular group on how species were delineated.
Okay. And philosophically, too, all my pens are disappearing because I'm leaving them there. Philosophically, remember what he is saying here is that special creationists were saying that, right? There's no connection between these in terms of creative acts. The creative acts are individual, independent creative acts. There's no connection. And what Darwin's saying is that, yes, they're connected, and in fact, over time, if we go from here to here, we have a succession of small changes and all the way up, okay? We end up with B, and we started with A. And his point was, well, where are you going to draw the line? And if I draw the line here for some reason, how is that different from here? So philosophically, he was always battling against this idea that species had some inherently important characteristics that biologically made them different from what he referred to as well-marked varieties. And well-marked varieties in here are somewhere along this too, but he was saying you're going to have a hard time telling what is a real, we would call a new species versus what's just a well-marked variety, not a species. What we might call races or subspecies or whatever, depending on what organisms we work on. Does that make sense? So this made philosophical sense that he didn't define them, but he really believed that they were artificial. He really held to that they were just, like I say, he said, they're just like genera.
So even today, talking about species, how we define species, species same problem. Like we don't know why 2% genetic change in a new species or 3%, why 50%.
Well, okay. So do you guys think we still have, you're arguing we still have the same problems, same problem in the sense that how do we define species? And of course, we're going to talk about what Ernst Mayr would say to that. Ernst Mayr would argue vociferously, if he was still alive, he would be very angry with this whole discussion and would say, of course we can define species. They're real biological units. Have you ever heard the term barcoding? Yeah, I mean, a lot of us, I'm not doing it, but a lot of my colleagues are. I think it's a very useful technique for giving us some information about biodiversity, right? You know, how much percentage change is it before we call it two samples different? Well, we have reasons for thinking there's a certain percentage in animals that there's a break maybe between species. There's some evidence for those genes. But then again, maybe there isn't. So I think you're right. I think we're still struggling with it to some degree. Yeah.
But there can be a point of no going back, right? Like irrespective of whatever environmental changes, two branches will not fuse again, and whatever changes happen then will be like there will be different species.
Mm-hmm. Yes. So we'll talk about some other species concepts. We stop at 1. Is that right? Okay, 1 o'clock. Probably we will not get much past Ernst Mayr, but we'll see. For this afternoon, we'll see how this goes. Right. So, in terms of evolutionary time, or evolutionary change, there's what I guess you're arguing is there's sort of a point of no return along here. But remember that's what Darwin was saying. Picking out where that point of no return is going to be the subjective part. His argument. Go ahead.
I agree with that, but that doesn't mean that we cannot say that there are no two different species because there are.
Yeah. Oh, no. And Darwin would never have said you couldn't make a judgment on how many species there were. He was saying that it was a subjective judgment made by experts in whatever group of organisms they were looking at. So he was saying that, say, in my plants versus his barnacles versus whatever you guys are working on, there will be different sets of characters, different traits, because he was looking at morphology, that are important, and you have to decide, as the expert in that field, whether or not the magnitude of difference between this group, this individual, and this individual is specific, or generic, or whatever else. But his argument was that it's not a biological trait per se that you're looking at. Yeah, it is, because it's biological characteristics, but the magnitude to decide you've finally reached B in here, that's what he was saying. And that's why his species concept, we could call it a morphological species concept, but he just emphasized so many times it's going to vary between group to group to group, that even calling it a morphological is probably inaccurate. Okay. Other questions, comments about Darwin? Oh, the other thing is, as we move through these papers or readings or whatever, if you remember a question you had, or think up a question you had on introductions to the different sessions or whatever, or we go down to Templeton number 5 and you think of something you wanted to ask about Darwin or whatever, we go back, okay? We haven't reached a point of no return once we say, okay, well, let's think about Mayr, you know. We can talk about anything on this. So ask questions as you're thinking about them. Anything else about Darwin's ideas.
Whenever we think about something like classifying species, there are always exceptions that we might observe, and I think that is why Darwin might have refrained from putting possibilities. He might have seen things he couldn't probably fit into a centralized model.
Yeah, I mean, once again, yes, he saw exceptions. Much more so than, say, we do a lot of times in systematics, plants and animals in particular, where my very first paper in 1978 named a new plant species. We had a type, we had a holotype, we had, whatever else, types. What does that mean that I had a type? And I wrote it up with my mentor. We described it. We measured everything. It means that to us encompassed the essence of that species, right? It's the Linnaeus system. And what that does is it artificially constrains, because we knew there's variation out there, right? We know there's a range of variation in that quote-unquote species. But I guess my point is that the artificial nature that Darwin saw was that kind of thing, right? Look, trying to define species is always going to be artificial, because we just don't know where to draw the line between, because remember, he's thinking about it, that long lineage. He's thinking about it, descent with modification. And he's saying, how are you going to know when to draw the line there in that plant versus that animal versus this, whatever it happens to be. So that was his point, is making that judgment is always subjective. That was his argument. I'm not saying that we have to conclude that. That was the conclusion, though, by the father of evolutionary biology. So we need to know where we're coming from historically, I guess. It's important, I think. It was important for my development to realize that. I never knew he didn't have a species concept, that he said it's all artificial. Blew me away when I first read it. And then I read it multiple times in other writings of his. He made his point. It's like, and he was fighting against creationism. He honestly was. Philosophically, that's what he was doing. He's saying, you can't divide them like a creationist. They don't have special, his argument was, they don't have some special essence about Iris fulva versus Iris brevicalis. They're different, but in terms of where they came from, they're connected.
Also, I suppose Darwin's explanations for this as being philosophical also was based on the observation of this, in the very first chapter we read, so variation and the domestication.
Yep.
So he did this pigeon crossing experiment and cows breeding. So this was probably based on those experiments.
Oh, I'm not arguing that it doesn't have incredible experimental, or didn't have incredible experimental and empirical evidence and natural evidence for his conclusion about this and about descent with modification. But the reason I'm pushing it on this and making sure you understand is because when we moved to Meyer called Darwin hopelessly confused multiple times about multiple things. And one of the things that Ernst Mayer's biological species concept, oops, I gave it away, is what came out of the neo-Darwinian synthesis and drove evolutionary biology for decades. And so we need to understand that Meyer, on the other hand, would be rolling around in his grave right now, again, with the idea that Darwin might have been right. That it is subjective. At some level, it's subjective. Going back to barcoding, I was asking people about this in China. Tons of them are doing barcoding there, which is great. But I was talking to botanists, and they said, well, the animal people have it easy because there's this break, certain percentage in the mitochondrial genes, blah, 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 and they really probably are looking at different species. And I'm like, how do they know? How do you know? Have you really gone out and measured the ecology and the reproductive potential and the blah, 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 blah under natural conditions? And the answer is no. It is a subjective call that if you have this much percentage sequence difference, you have a different species. And it's okay to make it. We just have to realize that's our assumption. We all have assumptions, right? I'll tell you about my species concept later, but okay. But no, I'm not, please don't hear me say that I think Darwin was on sandy soil with his arguments, because I mentioned that they're philosophical as well, because they were strongly philosophical, because I don't think creationism is a science, even though it was the accepted model at that time. So I think his arguments had to be somewhat philosophical too, or thinking about them were philosophical as well. But no, he had very, very strong evidence for his arguments, as had others before him for descent with modification. Okay. Great questions, great points. What else? Anything before we move on? Okay, so define the biological species concept. That's Myers. Or what is the biological species concept defined around? Maybe that's a better way. What does it depend on as to whether or not you have two species or one species? How do you know?
In essence, especially if they can reproduce together, they are
Okay, if they overlap in nature and they can mate or reproduce, they're the same species. So it has to do with reproductive isolation or not, depending on, in terms of defining species. Okay. How many species out there do you guys work on that overlap in nature that you know do or do not hybridize? Regardless of what you're working on, plants, animals, whatever. Do all of your species overlap in nature? Let's ask that question first. Okay. So, but Ernst Mayer's species concept is, I will know, he would say this, we will know whether or not there's 2, 6, 8, 10 species, whether or not they overlap in nature and can or cannot hybridize. So how do we do that? How did he apply? Because he argued you could apply his biological species concept to all plants and animals. This is a thought experiment. What do you think? How could you do the biological species concept? How could you apply that to a group of organisms that don't all overlap in nature?
As per the definition, they're still reproductively isolated for whatever reasons. So you can still apply that Myers definition. But if you do bring them together artificially, they might or they might not reproduce.
So Ernst Mayr, it's a good point. Ernst Mayr, though, argued for the biological species concept was actually or potentially reproductively isolated. And what he meant by that was geographic isolation didn't count. So we have to infer, so in other words, when I bring my plants in from wherever in nature and cross them in the greenhouse, and we know that they can mate, that doesn't negate applying the biological species concept, or sorry, it doesn't negate the fact that they may be different species. Because under artificial conditions, you don't have the niche, you don't have them growing where you don't have the pollinators, and so we're violating all of those, and so they may be able to cross them, but they may still be, if they happen to overlap in nature, they may still be reproductively isolated, or not. But what he's saying is that if you have geographically isolated populations within a group, there are ways of inferring whether or not, using the biological species concept, even though they're geographically isolated, you have to infer whether or not, if they happen to overlap, they'd be able to mate in nature. So he says you have to, he argued, and other people who have applied this absolutely argue that. Going back to barcoding, okay? Barcoding, let's just say in animals, there's supposedly this, I don't even remember what the percentage break is now, amount of difference. The idea there is there different species because they're 7% different in those genes or whatever it happens to be. The inference is they're reproductively isolated. It's still an application of the biological species concept. That should give you an idea maybe of how maybe Ernst Mayr argued how he could define in his day whether or not he was dealing with however many species. What do you think he would have done? Yes?
You could look for common species that were occupying both, and they were integrated.
Okay, so look for species that, sorry?
Were same species, which was in allopatry, which was in battery with both those different non-selected species, and then see if they were breeding.
We're getting there. And that's one of the ways that he suggested, yes. And so, whoops, wrong one. So it had to do with allopatric and sympatric or parapatric groups or distributions in a species complex. Does anybody know what Ernst Mayr worked on? What kind of organism? Whoop. That would be me. Hang on just a second. Does anybody know what he worked on? It actually has to do with the example I want to give you. He worked on birds. He was an ornithologist at Harvard, and he was a systematist, an evolutionary biologist, but he went out and shot birds and skinned and stuffed them. I did the same thing with lots of mammals. I'm not judging him. And brought them back to the museum and studied them. And he studied the morphology. Okay, so you said find, let's say, A overlapping with C, B overlapping with, let's say, D, and over here, let's say B, maybe like that or something. And so you look here and you ask questions about what these these different groups are able to do in these areas of overlap. Ernst Mayr went a step further, though, and what he said was if you didn't have this, which you basically could pair up and say, well, if this crosses with that, but this one doesn't, then what he actually did was he argued something like this. He worked on birds in Australia and in the Papua New Guinea area. So I'm just going to draw some things like this. So mainland Australia, you have hybrid zones, or overlap areas, I should say, between some of his species. And here's what he argued. He argued that if there's no hybridization between A and B, and you look at the amount of morphological difference between A and B, because those are the kinds of characters he had in the 60s, 40s, 30s, 40s, 60s, whatever. So you look at the amount of variation here, or difference between A and B, and you know that they do not hybridize. then you go out and you compare the amount of difference between A and B, and you ask if C, D, and E have more or less divergence from one another in A and B. And you can then infer, because you know A and B have that amount of divergence and they're not hybridizing, you can infer whether C, D, and E are reproductively isolated. Does that make sense? Problem was, Ernst Mayr didn't read, or if he did, he ignored Edgar Anderson and Leslie Hubrick's paper in the American Journal of Botany in 1938, where the term intergressive hybridization and integration were coined. And Anderson and Hubrick pointed out that you can't use quantitative traits like morphology to detect intergressive hybridization. Not past the backcross 1 anyway, and even in the backcross 1 generation. In other words, your backcrosses start looking like the recurrent parent, the parent they're being crossed into, and you lose all of your statistical power, if you will, to define that you actually have hybridization going on and integration going on, if you're using morphology alone. Ernst Mayr's birds on the Australian continent are now known to be integressively hybridizing a lot. It's just that it just doesn't show up in the morphology. So that inference was weak. But we're using that kind of inferential approach, once again, with things like barcoding. That's important to know. We're saying that a certain amount of divergence equals or equates to different species. Okay? Not arguing we cannot do that or should not do that necessarily. I'm just saying you need to understand that inference that's what you're inferring, or why you're inferring it.
Also, barcoding doesn't account for the uniform variation that occurs across the genome. Because barcoding, you also need certain genes that are assumed to be moderately evolving.
Mm-hmm. And you're saying that this doesn't take in. You're talking about the morphology doesn't take it into account, or just or barcoding.
Yeah, barcoding doesn't take into account all different genomic variables.
Right. Barcoding, the point is really well taken. So barcoding is looking at very limited amounts of data, limited genomic information. The whole point, though, of course, with barcoding, one of the points is to select genomic regions that will have, and this too has some really, probably some weaknesses to it, but it's selecting genomic regions that are not under selection, right? That's what you want. You want genomic regions that are not affected by, genetic drift either, but are not affected by natural selection. You want neutral mutations accumulating to a certain degree, which means that correlates with time since divergence, and so if they have that amount of divergence, then they've been diverged for a long time, and those are neutral mutations, quote unquote, so we're going to assume that the non-neutral mutations are really important, have accumulated as well, and so they're different species. There's a whole cascade of inferences from that percentage. I agree it doesn't take into consideration much of the genome, but I agree with that, but there's a reason for why they select what they select. But, yeah. I mean, to me, barcoding in some ways is triage. It's what I refer to as triage. We're doing the best we possibly can to get information from particularly areas that we think may be under threat. And we want to know what's there quickly and as quickly as possible so that we can garner support for conservation and things like that. And I just don't have any problem with that. I'm not sure if we're getting the kind of information we're hoping we're getting, but I understand what people are trying to do. Okay. does anybody here apply the biological species concept? You're not going to be judged. I'm just asking. Many zoologists do. People working on animal groups quite often are still trying to apply the biological species concept. You know, maybe relaxing it some, but do you use this? Okay, let me ask another question. What species concept do you use? Let me guess. I'm supposed to tell you what to use. Surprise! Ain't gonna happen. So what do you use for your studies? And maybe you don't have to in what you're working on, okay? The kind of questions. I understand that if you don't. But those of you who do, I'd really love to hear from you. We have a little bit of time before, a couple of papers afterwards, and I have some other things I'll share with you afterwards. But after lunch, I mean. So what do you do? Yeah.
Mostly morphology.
Okay.
Also use molecular data, and we have using the principles of monophyly.
Okay. What do you work on?
Plants, trees.
Okay. So morphological, sort of morphospecies, and molecular. A lot of us use this. I mean, I use a combination of things like that as well myself. Okay? But why not the biological species concept? I'll put you on the spot. Why not? If they all overlapped, would you use the biological species concept? And if they reproduced with one another, would you reduce them all to the same species?
How do you test the biological species concept? I mean, unless you're doing experiments, how do you, in the wild you can't.
Well,
In the field you can't.
Yeah, if you're out though in the wild and you have some of yours are overlapping and you genotype and you find that they're intergressing, you know, they're exchanging genes to some degree, you know, they're mating with one another. So my question is, with your trees, you know, you have these very different trees, but you know they're exchanging genes. Would you reduce them to B or D? This can be a question because we're talking about the biological species concept, and I wanted to actually make this as a general question too. I mean, wait, we can start with you, but do we do that? I mean, should we do that? If we know that they overlap, we now know that Ernst Mayer's birds that he loved and studied, that he thought weren't hybridizing, are and have been throughout their evolutionary history off and on. Should we reduce them to the same species on that basis, that they're intergressively hybridizing? Yeah.
Just if we look at it through time and see if it is, if they are collapsing in one species or if they're keeping it at a constant rate,
Mm-hmm.
then it could help us to decide if we do or not.
They're still differentiated morphologically. They're still differentiated ecologically to some degree. I mean, they're overlapping. They're not sympatric. We have many examples of this. We'll talk about hybrid zones and things like that later, hybrid zone theory. But the biological species concept, I mean, strictly speaking, states potentially or actually reproductively isolated, and those are not, right? Not really. I mean, they're exchanging genes.
Another way of looking at it is, even if they hybridize, how often the hybrids survive and themselves reproduce?
How often do they?
The hybrids themselves survive and they reproduce.
Mm-hmm. Okay.
If the hybrid is not
For example,
fertile, then we don't take it separate. Like if the hybrid is not fertile, then
Mm-hmm.
like who is calling separate species? If the hybrid is also fertile, then both are separate.
Okay. So how often do they, so basically we can ask the question, assortative, I will get to you, assortative mating or not, right? And the fitness of hybrids, okay?
Given the enormous amount of diversity in like pre and post voting supporting hybrids, and we also like species specific structures such as genetic area, which are also involved like provide anchoring to the species. So isn't taking all this account it actually supports? I mean,
So you feel like the majority of data would support the biological species concept or use of it. Okay? Yes?
About testing the biological species concept is, I think there's a fundamental law where if you're going to take two separate species from two different ecological areas and keep them in one, firstly, which one, you're changing the ecology of both of the species and you're asking them to mate. And reproduction, we know, is to a large extent driven by the environmental factors. Your entire endocrine hormonal system is driven by that. So it's not natural selection anymore. So you can't say that is how it works in nature. It's becoming an artificial.
Yeah, so we can consider, so is this artificial? I mean, these are what we're, this example, what I'm saying is these overlap in nature, right? I mean, they've actually expanded their ranges, if it's secondary contact, expanded their ranges and now are overlapping with one another. So,
You can look at the frequency, like was mentioned,
yeah.
frequency of the hybridization.
Yes, but what I also want to say is your point's a good one, because some people would argue, well, because it's at the margin of the ranges of both of them, it's not in their
It's more
quote
of a
unquote,
generalist species more than rather species.
and it may not actually be in the center of their realized niche, if you will. So this could be like living in a disturbed
It's
habitat.
almost like a third species of
Ecotonal regions, that environment genotype kind of interactions, yeah. And others have pointed out that okay, well, what goes on in the zone, yes, stays in the zone. I know, but I mean, that's what they are arguing basically, is that what goes on in here isn't that big of a deal. It doesn't tell us about species. The problem is that these are moving across the landscape in very rapidly relative to evolutionary time. So they're actually, it's not as simple as we have this zone and it's always been there. And genomically, we know that throughout these genomes of these birds and other things, there's actually integration or transfer of material from here into here, however it happened. So this is a porous zone, especially for adaptive traits to move across it. So it's more complex. Maybe if I could say it this way, is that application of the biological species concept or Darwin's concept, lack of concept, we actually have to, in many ways, relax the species concepts some. Okay, not have them hard because it's biological now, or it's a biological system. For example, if the hybrids are able to back cross to the parents and you have integration, we ought to reduce them to one species. That is actually the application of the biological species concept, right? The problem with that, I'll give you one example. One example of a problem with that is all dog-like stuff. Gray wolves, red wolves, coyotes, domestic dogs in North America would all be one species. These are incredibly divergent forms, morphologically, genotypically, genomically, behaviorally, okay? Very, very different kinds of animals. No mammalogist and no canid evolutionary biologist in their right mind would suggest that we do that. And yet, in some zones, we have 30% hybrids. That's huge, right? So if you see, we have to be, there's art to this. There's art to the, no, you hate hearing that, art and philosophy. I thought this was a science lecture. Or discussion, but art and philosophy, there's an art, there's a subjectivity to how we apply these kinds of concepts to our group. Just is. If you want to be just the biological species concept, I can guarantee you that if you're working with group of organisms that ever overlap in nature, they're going to hybridize and integress some plants or animals. It's just going to happen. And so you have to decide how much of that, how much assortative mating do we need, how much integration is too much, if you will. Of course, I'm interested in the process, so it's never too much, but understandably for systematics it is. Okay, other questions or comments about Darwin or Meyer and their species concepts and their... Ernst Mayr really was very upset with Darwin. He took him to task a lot. But Ernst Mayr took a lot of folks to task. He once quoted, well, they asked him why he named it biological species concept, and because he said it's the only real species concept out there, and so we called it biological because everybody else's are non-biological. It's like, I've said some really arrogant things over the years and written a few. Never got there. I wish I had. And maybe somebody would quote me, oh, God, I can't believe he said that. He also said, they interviewed him later in his career, and they said, do people ask your opinion on speciation? He said, not as much as they used to, but all thinking biologists agree with me. Wow, cool. So, but I don't agree with him in a lot of things, and I've said this already, but you realize that he, in this course, is the most read author in terms of these readings, because we're not only going to do this, but session 3 we have another Meyer paper. The guy was foundational in evolutionary biology and incredibly intelligent. Arrogant as the day is long, but that happens a lot with a lot of us. So, great. He really, I've read, his 42 book is magnificent. His 63 book is even better. And like I say, I don't agree with a lot of things he wrote in there, but just unbelievably brilliant. I will nowhere near be as brilliant as he was. I think he came to wrong conclusions, but it may have been his data. All right, I'd suggest go ahead maybe and break before we go on into this and then something else I want to tell you about today. So lunch, I don't know how that works.